some technical centers. That's not going to happen. You need to build a scientific story. You have to bring leaders in science. So that's why you have to, we have to follow a certain core philosophy to how they're following the same track. And if you have more time at Barton, he has been doing a kind of microbiological part as well. So with this, I start my talk. So today I'm going to talk about this carbohydrate metabolism. I used to always fail in carbohydrate metabolism because all the TCS cycle nobody remember, but I learned a little bit. So, and then I'll talk about altruism, stem cell kind of a theory proposed first by Darwin himself about group fitness versus self-fitness. So this is nothing about spiritual altruism though. So I think considering that you guys are pharmaceuticals or even I will go maybe more about molecular a little bit, but I would not go deep. I will just tell a story about how we have done science to encourage you that you can also follow your own path. This is my identity a little bit. I'm a weaver, silk weaver, Muga silk weaver. I used to come to our Dibrugarh to sell and buy Muga. I still do it. We have a Muga silk museum. So we are 6th generation Muga silk weaver. So we're giving this free treatment of cancer in Suhakosi, Dibrugarh, Tinsukia, Margherite, our main center of Mugaso. So that's our telemedicine center, and this is one of this beauty of that even though I have moved out, but because of technology I can still connect it, I can still study, and this is I'm showing a survival of our patient data coming from us. This should be included in Partha's PhD, of course, and this was all done by Lakika Partha and our other student Titimoni Bosho for last about since 2018 this track. So you see those were cancer stem cell defense mechanism, those have the phenotype, they rapidly die. It's very difficult to treat them, and as you see that the pharmaceutical science is all about drug discovery. So even though we are lagging behind US or China, but the good news is that many of the disease discovery is still not met. Cancer is one of them. We have treated this cancer patient with immunotherapy. As I speak here, I have 2 patients, one from somewhere near Dibrugarh Tea Estate. Where is from Solomon? Remember?
That is Doigu,
Not Dibrugarh. Okay. One of the patients, he had lung cancer, stage 4, almost died. I started immunotherapy. He's living for last 2 years. So there's some good news, but immunotherapy also fail when there's cancer stem cells. I'll discuss about what is that. So there is recent news that a Chinese firm has developed a immunotherapy. Is a LD1 inhibitor. We call it nivolumab. Very expensive here. For 1% for 1 year, nivolumab will be about 15 lakhs. We're buying that medicine from America. A Chinese company, a small startup, has started developing antibody. Have gone through phase 2 trial just 2 months ago. If you buy that medicine now, you have just cost only 1 year supply 2 lakhs. If we would have done our biotechnology training policy in 2001, we could have saved so much of money from India, for India. This is the stupidity of our Indian policymaker, that they lost the whole game. They got really fixed with developing generic medicine only, oral vaccine, whatever. But they didn't realize that this nivolumab, through nivolumab, one drug, America is earning yearly $120 billion alone, I think from India, because India's cancer patient is serving. If they would have spent $5 billion in 2001, they would have saved so much of money. Now China is getting all the market now because they invested in the students like in 2001. Anyway, so this is a clear, real picture I'm showing you. Before I show all our molecular biology data, that how bad it is, this cancer disease now. It is becoming a lot of it's a big public health concern now. 2 months ago, one of my interview was published in Assam Tribune, if somebody read, and we showed that in our Sualkusi area where I have data, kind of as a community oncologist, I have data for last about 30 years, there's 70-fold increase in cancer incidence. It may be because we have enhanced diagnostic, whatever, but we are spending money to treat our cancer patient. And this money is going to drug manufacturer, not in India. And this is where biotechnology is going to play a big role in the future. Because without biotechnologists or good pharmaceutical guys, we cannot do any pharmacodynamics, pharmacokinetic studies. We cannot bring a drug from lab to phase 1, phase 2 trial. So this is one example of why we need to young guys need to knock on the door of this, all the stupid guys of DBT ICMR. They're sitting there missing the opportunities. They're still saying they're big. When they know they missed the boat. Anyway, so this is our computational laboratory. And we have a team. I'm going to show you a little bit more. This is started in 2010. Then this is our Thor lab. I moved my lab from Forsyth Institute at Harvard in 2018 because there's a class of intellectual property right. Harvard didn't want me to stay with Indian lab, computational lab. They said whatever I do, it belongs to them. Indian government said, if you don't quit from Harvard or something, we would not give you zero certificate. Scientific and industrial research organization certificate, we need it to actually submit grants. So we quit. So we have an independent 2 lab now. This is one in Lowell Institute at University of Massachusetts. And then this is our mission of computation. So this is the whole story actually. That India has a great heritage of scientific progress. It happened a long time ago. We have this Nyaya Sastra methodologies. How to do scientific experiments. All were given in Nyaya Sastra. And the first experiment, scientific experiment was documented actually in Rigveda called Aruna. So we have methodologies and we have ideas, and then how can we transform those ideas, philosophy to science? So we had this debate in 1992, 91 in our village, Sualkusi, with Malanya Asarja Hembhai Bab. Hembhai is a big Gita scholar, everybody knows him probably. And then he's a Vedic scholar, and then Malanya late Dal Krishna Bora. He has been a mentor of Rashtriya Swayansemak Sangha for many years. He passed away recently, and my dad, Krishnaram Das, was a poet, and they all work on the philosophy of the avatar. When during stress in the society, how somebody from this one human being like Krishna Ram transform the godlike power. That was actually philosophy was done avatar in Pancharatra Tantra, Satavada Tantra. And that philosophy was the pioneer of constructing this Hajas Hayagriva Marat temple in 6th century. So our mandate has been to transform that avatar philosophy, apply that philosophy to study stem cell or study cancer. I'll tell you how it goes, the link. And this is my talk, actually started something we first presented in Milan, Italy, in 2005, about this the tumor stemness switch. So at that time, cancer stem cell idea was just beginning. And I gave a talk there. And since then, we scientists always tell our stories, keep some slides always, intact. So I'm going to talk about these two ideas. One of this stemness. Stemness is this about identity of a stem cell. And you like it very much when I tell you how stem cell has intrigued me or many other scientists. It is all about identity, self-identity. We all strive for our core identity. Who am I? And we always look for a path. You guys are trying to decide to become a pharmacologist because there's something inside inner call. Maybe it is going to come future, but most of you are in a situation where you cannot decide. This is called uncommitment. But you have the potential to become anything. That's stem cells. Stem cells remain in an undifferentiated blood. He thought, and his team, that if we can find out how a hematopoietic stem cell become RBC, we can make the blood in the lab. That dream is still not fulfilled though. I mean, some billions of dollars have been spent. R.F. Weissman was a Stanford Stem Cell Institute director for many years. Now my friend Robbie is their director. They have funded so many companies just for this. If we can develop artificial blood, I mean, we're a billion. Maybe now, whoever does will be a trillion company. But it's so complex. I have been digging one pathway, molecular pathway called VEGF receptor 1 that I identified in 2005. One pathway HIF to few proteins. The more you go inside, more deeper mystery goes. However, we need a lot of pull. We need a lot of talented students to be in that. At that time in Toronto, in Boston, in America, we had that starting from 1980 until about 2010. And now American situation is shaky, even Europe too, because they don't have talented pool anymore. Now it's China and India. So I'm just telling you, the stemness is about that. And now think about that. Cancer has somehow hijacked that identity of stem cells. It's so much complex. So no matter what kind of medicine you give, the stem cancer again come back. And if you have any dear one, loved one who have cancer, you find out that patient is cancer, this is bad. Even sometimes for 15 years. And if you take the cells to the lab, I can guarantee you will find their cancer stem cell, they remain the same percentage. If you have a primary patient has cancer stem cell population in the head and neck cancer, 2%, no matter what metastasis, what happened after 10 years, 15 years, when the patient return back to your clinic, you take the tumor cells, put in the lab, and look for the stem cell population using a marker, say 2 or 3%. They're so consistent. So we call it self-identity. In fact, we have published a paper in Frontier Immunology just 2 weeks ago, that Lekika's first authorship paper. We have redefined this tumor self-identity. And this is where we're sticking. I am sticking right from beginning, because if you know that about Hindu philosophy's main core thing, ontology, we call, is identity, self-identity. So it's So Hindi philosopher has spent thousands of years asking this question, who am I? The being. We are science through stem cell, we're asking the same question in a molecular biology perspective. We're asking the same question also for cancer stem cell. And that's why the stemness is so very important idea. And this is about now kind of the core slide I'm going to present. This is basically sum up all our work, right from my 2005 paper in cancer research. So those who know this journal, Cancer Research, this is one of the key academic journals in our field. In our time in 2004-5, it was our journal. So it's very difficult to publish in the journal. Now there are so many new journals come up. So it took us 2 years to publish the first work. This identifying this VEGF, vascular endothelial growth factor, and its receptor 1 signaling pathway, autocrine loop. We identified that's also in hematopoietic stem cells, and we also identified in cancer stem cells. Now, like if you see the slide here, the cancer stem cell, out of the millions of cancer cells, those cells, cancer cells that can give rise to the tumor again, that can cause relapse, we call them cancer stem cells. So they can reprogram means becoming a different being, different state, to a, we call it altruistic cancer stem cell. I'll explain it later on. More powerful, aggressive. And we now have a kind of a diagnostic biomarker to identify in our patient when this transformation happened. Like right now I have a patient of multiple myeloma case. Right now actually I'm speaking, I'm texting over WhatsApp what to do. She's now in Health City with my friend, medical oncologist, under treatment. For 2 years she was doing very well. Doing very well. And 1 week ago she came, her son came and saw my mom is feeling weak. Not very energy. I said send a video, 15-second video. She was cutting, vegetable, but her eyes were a little shaken a little bit. So we have some test, we did it, and I said go for the blood test. So CBC, shows there is peripheral blood has 25,000 white blood cells. Normally should not be more than 11,000. I thought, okay, maybe infection. Infection can increase neutrophil count. But then you look for, we found 81% blast cells. That means 81% of our blood cells are all blast. Neutrophil only 5%. Lymphocyte only 2%. Anytime she's going to die because of infection or bleeding. Her platelet count was only 5,000. 1 week, everything flipped. We have to immediately hospitalize, do all kind of tests. So this is now in our lab. We take her blood, and we do this all kind of tests. And then when you look at our cells, we know okay, she has this tremendous inflammatory phenotype. Those cells secrete a lot of inflammatory cytokines, IL-6, TNF-alpha. If you do the C-reactive protein called the C-reactive protein, CRP we call it, 250 something, it's been crazy. And most of the patients die. And we cannot detect them. Medical oncologists has a hard time worldwide because most of the medical oncology practice is done in the big hospital. There the patient goes only when it is this crisis. Even in Mass General Hospital and Harvard Medical School, same story. So I thought that I need to continue with my community oncology practice. I can follow up the patient life history so we can detect them. So So that's why what we do our science has more confidence. We're not only sticking to lab. And this is the problem in India. In India, there is no MD PhD. All our doctors are busy seeing patients. They're in a cycle of earning money and drinking more wine. Because there's frustration, there's no feedback. They do not know whether their treatment is working good. They don't have a good scientific team with them. Ideally, these pharmacologists, pharmacists should have been working aligned with our clinician. This is called MD PhD program. China has implemented very well from 2001. Indian MCI lobbied in our Medical Council of India, one of the corrupted organizations in the world. They lobbied, oh no, we don't need PhD. They're not trained. They're going to take our job. PhD said, forget MD. They do not know anything. So there's a whole rift, ego class. Otherwise, that's why we don't have confidence amongst clinics, what they're doing. So they're following American protocol to treat cancer patients. Americans, we're getting money. I don't mind that. I'm also American, so why not? Sell drugs to India. And our scientists, they're going on a small scale industry job, something in Bangalore or Delhi. There's no confidence of the data because there's no testing. So that's why it's always important to have a setup like what we do. So that's why I thought I need to set up this co-educational lab in a small corner of India, in Swankosi, where nobody disturbs us, no politics. So we can continue to develop our data and then started showing up. Seniors, nobody in Assam knows I have a lab. If I tell you we have all this publication coming, we have made a groundbreaking research work that bacteria goes inside stem cells coming right from Assam. Nobody in Assam can tell them, including Health Minister of Assam. Now he's just here. Nobody. Because we did not want to be disturbed by politics. So now that we have done it, I'm coming for outreach. That's why when Sandhya said, come to Dibrugarh, why not? We can tell our story. So the story is this that you need to have the confidence about science. You need clinics and also you need this, the scientific lab. This institute was set up in 1983. At that time, I'm telling you, John Dick, my former mentor, was dreaming about a billionaire. I'm pretty sure somebody in 83, 85 was dreaming the same thing. Because I really appreciate that our Indians, we have talented students, also in Assam. I'm from Assam. But because of the political crisis, political lobbies, we could not fulfill the dream. Because we always rely on politicians to do our job. That was the mistake we have been doing for a long time. But we cannot do that because they do not understand. If you tell what is biotechnology to a minister in Assam, nobody understands, I'm telling you. So this is why for you, I thought this talk should be about life journey of science. Okay, this is the scariest slide I'm telling you. I'm not going to talk about that, but this was my title. Carbohydrate metabolism, if you see, is an evolution. It is one of the early metabolism arrived for. It's simple molecular glucose. How it become an energy producer. Also, it produces a lot of nutrients, like say the glutathione as an antioxidant. Glutathione has a history of about 2.5 billion years, right from archaea. It has not changed. It's tripeptide. Then we have the pentose phosphate pathways that lead to NADPH, nucleotide components, hexagon pathway, all kind of protein glycolation. Many of these pharmaceutical sciences heading for that, nanotechnology, whatever, there's hexagon pathway, and then a serine glycine biosynthesis, and so all those that this years ago we have studied and we're still trying to study. And I'm going to kind of give you a little bit picture about how stem cells use it to live in a hypoxic environment. Hypoxia means low oxygen. You may be surprised, well, I mean, why low oxygen? Well, all our stem cell niche, like bone marrow has this niche, the hematopoietic stem cells resides in this low oxygen, only about range from 0.1%, almost anoxia to 3% oxygen. Bone marrow is designed like that, where some of the stem cells will stay about 100 micron to 150 micron away from the next capillary. Why? Because if they have any hypoxia, they can follow a Warburg effect, or what we call the anaerobic glycolysis. They're not going to use glucose for kind of mitochondrial biosynthesis or ATP production, because that leads to a lot of reactive oxygen species production. And that can cause genomic damage. So these guys, the stem cells, that's why they reside in low oxygen area, where there's no oxygen. And oxygen is a double-edged sword. It can sustain life. It can also damage the DNA by causing oxidative stress. So, and also hypoxia, we all are living beings, and all our initial life come from low oxygen, no oxygen. Bacteria, cyanobacteria, archaeas, they evolve without oxygen. I have a book written also about that. So that carbohydrate metabolism is so complex. You can all graduate, you can have one lab for each pathway actually. That's what China is doing. That going deeper into it. And you need to have a model like we have altruistic stem cell, we can have any kind of cellular model. But I'm telling you, if I want to say which lab in Assam is or India is focusing on carbohydrate metabolism, maybe one or two, not many. So I will briefly talk about that. And these are our already graduates from our lab. Now Zoita, she is doing her postdoc, she's ISM fellowship in AIMS. She's still working with us. Zoyasri is now, I sent her to Stanford University to continue direct my BSL3 facilities. And she did her PhD with us from JNU, because when I say they do PhD with me, I organize an arrangement at universities and IIT that I would be external guy. All the research done in my lab, under my direction, and officially there is a, what they call, supporting PhD guide. Lekika, what is the name in IIT? What they call?
External and part-time.
No, I know. What is Sanjipta's name? What is her role?
Okay. Coordinating supervisor.
Yes, called the coordinating supervisor. We actually tried to do that at IIT so that it could be implemented across India, so that R&D guys, like I don't have time to teach, right? But we also need to be part of this training next generation scientists. And in India, that model is not there at all. I have to fight with kind of JNU to have that, to have the history of fighting or continuing fighting with Gauhati University to have this rule defined. And finally, IIT, they accept that. We have the coordinating supervisor. And that's a very good model where a research lab like us, who can train the next generation biologist scientists without being engaged with this full-time teaching. Those the faculty, you have to teach. You don't have time for research. So they have to do some combination. That's what we did. And so we have here also Lekika here, and Ibrahim, he already completed. He's now in Saudi Arabia. So with that, how many minutes they go so far?
15 minutes.
Another 15, 20 minutes, I'll talk about real science now, okay? So far I'm talking about this, how to say, introduction. Now, next 15 minutes, I'll talk about science. First of all, altruism. So altruism is about group fitness. The bacteria was considered as a unicellular life for a long time. And then in 2012, a lot of awareness came up. The same year when we published our discovery of altruistic behavior of stem cell, there was a recognition that we need to also consider bacteria as a social organism. That they have, we call this kind of a society, about group intelligence, about biofilm production. That recognition came because many bacteria seem to lose their fitness to enhance group fitness. And this altruism was actually also very much studied in animal kingdom, mainly in honeybees. So if you consider the honeybees, like, most of these bees, they sacrifice their reproductive fitness just for the queens of the bee, So it comes from altruism. So from there it came to bacteria. But one of the key thing here, altruistic, this biological altruism doctrine, first proposed by a French philosopher after Darwin's contemporary. Very difficult name, I always forget his name. Darwin's name is easy to remember. But Darwin also accepted that certain form of group selection might happen. But molecular biologists and modern new geneticists who do not agree, or those that do not agree with that. So altruism is still a very controversial theory. Because how can I explain the selection of this altruistic gene? I mean, if somebody is sacrificing their life, they're gone, right? And then a cheater will take advantage of that. It's called a prisoner dilemma. Okay? If there's an altruistic prisoner, the enemy will plan everybody to escape. He'll be the last one, and the chance of him getting caught is very high. The cheater will escape. So that eventually the cheater will repopulate the whole population. So that's why it is not being accepted. But in certain conditions, it could be possible, like for example in bacteria. We have the quarantine, the words from, I forgot the name, French name again, biofilm. But we still do not accept it because we have not been able to demonstrate that the intention was right or wrong. Was the intention really sacrificed or they have been forced? So intent is the important thing. So then you can imagine that for us to study stem cell altruism could be so much controversial. But we have been stick to that because theory in science is all about trying to explain something which we don't know. Maybe our theory might take 200 years to prove. But the key thing is that we do not know how to explain many of the drug resistance mechanism of bacteria, even by molecular biology, even by Darwinian selection process. But some of this phenomena can be explained by bacterial altruism. For example, E. coli resistance in a dish, there could be continuous selection, but then from then, few of this bacteria always persist. They remain sensitive to E. coli, to the antibiotic. Even though they're sensitive, but the patient is dying, or they can actually still grow. So that's how, in a very controlled experiment, altruism has been explained in bacteria. But our altruism, we talk, is something different. Because we recognize that initially, that altruism to have, there have to be a self-fitness gain. If I want to sacrifice fitness, but I am a poor guy, like a fitness, who cares? I can say, to be honest, to be like, become popular. Yes, I could have been rich. I did, but I sacrificed my life. Well, it's better to be very rich first, gain my fitness, and if I sacrifice then, and then we can believe that. So the biological altruism proposed to Darwin is about basically a honeybee model, where those who are sacrificing, they're already poor, already weak. Whereas in avatar, we call jibo upakar. The theory is that one has to be very fit, sacrifice, and then that person has to disappear. For example, Krishna had to die after the end of Mahabharata war. Even though he could have easily defended himself from this arrow from a hunter, he told Arjuna, my time is up. If I stay here, it would be really bad for the world. Similarly, same thing happened to Ram or any avatar, Narasimha. He was asked by Prahlada, why don't you stay? I mean, these are mythology, but through mythology, philosophy was explained. And this philosophy of avatar was even then taken by Buddhists to develop their Bodhisattva philosophy. Say, for example, in a Buddhist Karana Vyahushidra. By the way, I stayed in Bhutan as a doctor for 4 years, between 1994 and 1998, so I had learned Buddhist philosophy as well. So there they have really talked about that. Bodhisattva had to be very powerful, go first to heaven, then come back to earth. Or do not attain nirvana. Sacrifice self-nirvana to work for the society. So in that context, so our altruism was first described in a poem book called Honalina Khorju. It was that Daman Nirjatan Briddhi Hai, Hottakahi Prahaladar Hridayar Jitu. That was our foundation of our theory of avatar. That more there is a stress, more the inner power comes. Now, when my dad had cancer, so he asked me to start this research that there should be some avatar cell in our body flowing in cancer patient that's going to fight against cancer. So he refused to get treatment, but I took him to AIMS anyway. At the time I was in AIMS because my uncle is Dr. Bhavananda Das. He is a famous heart surgeon. He said, bring Kura to AIMS. So I took him. So that's how my medical oncology practice started. He was my first patient. We came back to Sumalkosi. I started my clinic and a lab. So then I realized, I did not realize about this importance of this philosophy. I thought this is crazy. But when I was treating cancer patients or all other patients in Bhutan, I had this interesting insight while doing bone marrow biopsy. For many of these patients would call pyrexia of unknown origin. So we do not know the cause of the fever, then we do bone marrow biopsy. There we found a bacteria inside hematopoietic cells. Many of our TB patients come with fever. We cannot find the TB inside the lung. We call them extrapulmonary tuberculosis. So I then suspected bone marrow might have TB, and then it took many years, but that clinical findings sparked my research area on this evolutionary role of stem cell lineage defense. Because the question came to me, well, if the bacteria can go inside the stem cell lineage, there must be defense mechanism. Because they live in hypoxia. By the time we had the PubMed always coming, so I knew about the war was going on. That question was, that sparked, okay, what is the defense mechanism in stem cell? So that, what's happening? This little frozen or okay. So then I wrote a book called Science Behind Squalid. Yeah. So here is a book you can read online, and this is about I kind of reviewed literature on this evolutionary life of unicellular bacteria, the antioxidant pathway. Squalene is the antioxidant. Is the intermediate of squalene is the intermediate of the cholesterol metabolism. You guys know in the pharmaceutical science has a huge progress when the statin was discovered, inhibitor of H-sensitive coenzyme reductase. Statin is now about $400 billion market by statin itself. So anybody would have from 83, our Dibrugarh would have come with that kind of discovery. Again, we missed the chance. Anyway, so squalene is also found in our skin. Many of our girls who are here, if you go to the cosmetic or makeup, look at the ingredient, squalene is there. And squalene was as a layer in archaea, primitive bacteria. And we propose that it might have a role in UV protection because life started in Earth with extreme radiation. So I was always interested right from beginning when I was with my dad in Ames, New Delhi in 1994. So why this cancer doesn't come back? I used to listen to many seminars in New Delhi. One of the seminars I remember was on this radiation. The cancer cell radiation resistant. And that makes sense when I was in Bhutan. I thought, okay, if they're radiation resistant, maybe they're hijacking some unicellular life defense mechanism. That's why I got interested in squalene. I think give you. Yes. Yes. If you see this is the diagram I had. I proposed the stem cell niche defense in 2000. So I went to this one of my professors there when I was in fellowship, clinical research fellowship in Toronto. I'm an MBBS doctor. There I did this MD kind of exam. And when I said I wanted to do research, I was not allowed because I don't have the background. I was said you can do this kind of MD in oncology, medical oncology, but you cannot do this medical oncology research. You don't have the background. So I wrote the book. I had this idea. I took to my professor that I am doing research on my idea. So he was so good. He took me and he mobilized whole thing that, okay, you have an idea, you wrote a book, you are in. He had the power. Believe me or not, all the faculties here in India feel powerless. If you find a good student, have a good idea, can you do that? No way. This is why you need to have the power. Faculty need to be powerful. Government have to trust in the faculties. But government does not trust our faculties. Government does not trust themselves. So this is the problem. If this professor did not allow me, I would not have been able to do that. His Professor Herman Yeager, Sylvan Barisel, and also Gideon Korn is the main guy. Anyway, this is the first idea. This is a naive idea, right? I just wrote it down, but I gave a book, and he said, well, it was a book by a medical doctor, and I got a company from Hong Kong, so who attracted whom I thought. I thought I need to also be business-minded, right? I'm from Swan Coast, always business. Important. I said, squalene, antioxidant, look for any market. There's no market, but there's a company in Japan trying to sell squalene as a dietary supplement. There's a business in Hong Kong too. So I made a contact. That, hey, I can write a book. I wrote a book, but you have to sponsor. They thought, well, if this guy's at University of Toronto now, doing a fellowship, lure a truck, sponsor. And then I put my idea in the book. Even the company said, oh, we need only just 20 pages. I made a 200 pages book. I put my ideas, because my goal was just to get to the PhD program. So you guys also have to do that kind of ways to navigate situation. Cannot only doing science in the lab, have to also have some politician kind of mindset, how to navigate. So this was the idea, and it took many years, 12 years, to actually prove the idea. Millions of dollars has been spent on that, to have published this, and first only, they allowed me use only one word there, stem cell altruism. This was published in Stem Cells. It was rejected by many big journals. Stem Cell at the time was a big journal too. But key point is this, that it's such a controversial area, and we have been able to sustain our funding for 12 years. In India, if you do the basic research for even 5 years, know any product, we call Puria, All this exhibition they do here, we call Puria exhibition. There's some herbal medicine, something, some product. India stuck with that. But you cannot do like that. If you want to develop R&D product, you have to spend millions of dollars in one idea. Yeah, either accept it, move on, or just say that we do not want to do science, or we do not want to do pharmaceutical R&D development. It cannot be satisfying with showing minister that there's a product here, herbal medicine. Like this convention happened, what they call biotech, as some advantages. There is no product actually that come from deep commitment from anywhere in India. That's why American, Stanford or Harvard, anywhere, they say science does not exist in India. Our policymakers get really upset about that. But this is true, what they mean science. Is any core idea came from India in the last 100 years after independence? No. Why? Because we need funding. Anyway, so this took 12 years, and this is our key finding. As I said, if you want to talk about altruism, you better come up with a molecular mechanism. Otherwise, it's impossible to even talk about that. So what we found was very interesting, and I'm going to spend about 2 or 3 minutes on that. So this is a human embryonic stem cell. We exposed to an extreme hypoxia followed by reoxygenation. We call it kind of a oxidative stress or hypoxia reoxygenation. If you have some loved one who had a heart attack and died immediately, this is what happened to the heart. The blood vessels suddenly stop flowing because of the blockage, causing hypoxia. That's not a problem actually, but immediately after a few minutes, blood flow started restoring. When oxygen comes back flowing, there's a huge production of a burst of reactive oxygen species that damages this part of the heart disk. If that heart disease has a conductive mechanism that causes ventricular fibrillation in his bundle, and that causes the patient to die before reaching the hospital. So this hypoxia reaction is a potent injury. And think about stem cell. The stem cell has a high p53 system, very sensitive. And p53, I think you all know, it's a protein called guardian of genome. Any kind of genomic damage, p53 is up. Either the cell is going to die through apoptosis or cell cycle errors. In case of human embryonic stem cell, it is always death. Because embryo cannot, any kind of live embryo cannot permit genomic damage. Then life cannot be sustained. The self-identity of life cannot be sustained. For example, your face and the face of a human being 60,000 years ago would be almost similar. This is called temporal identity of life. Like fossils, shark fossils we know from 350 million years ago. Same like today's shark. This identity of life is maintained by maintaining genomic integrity of this embryonic stem cell embryo. This is why p53 is so high in stem cell. Wildlife always ready to act. And in that case, we exposed those human embryonic stem cells to extreme hypoxia followed by oxidative stress, so that all cells die. We're looking for some stem cells to survive, hoping that some of them will survive by somehow altering p53. It took us 12 years because we almost failed always. And finally, using a specific marker called SSEA3 and ABCD2, we were able to find a tiny population of cells that upregulate HIF2 alpha and downregulate p53. And we found this is the core data. So what we found is very interesting. We found that p53 only remains silent for 2 weeks. And then we apply system biology and other proteomics assays. I will not detail. But if you see that even though they have this GCFSDA very high reactive species, on a day 4, the cells survive and their HIF2 alpha goes up, p53 goes down. But after day 16, the p53 goes up. So p53, as you know, those who knows about this molecular biology, is a negative feedback system between two proteins, p53 and MDM2. p53 goes up, MDM2 goes down. MDM2 goes up, p53 goes down. They have an oscillation. The amplitude and the strength of this wave with the oscillation decide whether a cell will undergo apoptosis or cell cycle arrest. That has been known for a long time. Even there are specialist labs just working on this negative positive feedback loop in biological system. And that has been now actually employed to develop artificial intelligence program called bioengineering, or based what they call the AI system. The decision making process is based on oscillation. So what we found that those few embryonic stem cells, there the p53 is transiently altered. So it's arrested, oscillation is arrested. In a low p53, high MDM2 state. After 2 weeks, because amplitude, the oscillation is usually about for 2 hours, it can go to about 2 days. It's kind of vary. But in a particular embryonic stem cell where they survive, they had 2 weeks of this oscillation got stuck in a high MDM2, low p53. And that allowed that stem cells to survive despite genomic damage. And these cells that secreted glutathione. Usually glutathione is not secreted by cells. They cannot afford to secrete their principal antioxidant. But these cells secrete glutathione. They also secrete squalene that we later found out. And so that allows this embryonic stem cell to secrete factors, antioxidants. And then when you take the conditioned medium of this cell and put in another embryonic stem cell exposed to hypoxia reactions, we see that a lot of fitness gain. Many cells survive. So not only glutathione, maybe there are other factors, but glutathione is the main one. So that's led us to hypothesize that, well, this is a fitness gain by the embryonic stem cells. Then we spent about 4 or 5 years just to convince ourselves or to show, demonstrate that they either become cancerous cells, because this embryonic stem cell, now that they have deficient p53, should be cancerous, right? because cancer is all about low p53, genomic damage. If they become cancerous, then it cannot be altruistic because fitness gain remain persistent. So we took a lot of time, we did a lot of studies and found that none of this human embryonic stem cell became cancerous. The system is so robust, the human embryonic stem cells, that after 2 weeks, the oscillation, once p53 goes up, the cells differentiate or die. So that became our first proof that there is this altruistic behavior. There are certain stem cells actually molecular, there's a molecular mechanism. And then we debated that this p53 probably, because this is about 1.1 billion year old molecule. And guys who are doing pharmacy, this is the big science about this, looking for kind of phylogenetic study of all the pathways, molecular proteins, because when you go deep into the pharmacology, you have to know about protein binding, 3D structure. You have to know the whole of phylogenetic. If you know p53 is such a dynamic protein, we have not been able to find a good inhibitor or activator. So dynamic. Anyway, so that means since then probably it is preserved. That was our hypothesis. That's why we allowed to publish. Okay, I think here's we have developed a mouse model that can show that we are found altruistic phenotype using a viral infected mouse model. I would not talk details about that. What happened? Something. Okay, we have to, I think I'll go skip, okay. Now, so that was our stem cell altruism, and then we have time, then we have two models developed. Sonda has a model where she is developing a mouse model of carcinogenesis to study whether the stem cell altruism is a big bang of cancer actually. We have a 4N2 induced oral cancer model, and Sonda has used that model to identify herbal medicinal product that can target a cancer stem cell exhibiting altruistic phenotype. So this is what stem cell altruism, as I said, it took many years to demonstrate that there is a fundamental molecular mechanism that can explain altruism in stem cells. p53 MDM2 oscillation. Now this could easily explain now why cancer is always about p53 deficits. If you study any cancer, p53 is always deficient. So we believe that that's how our study of stem cell altruism, discovering a deep molecular defense mechanism, we call it defense mechanism, because altruism is in fact a defense mechanism to maintain group identity. Coming back to this initial thing. So if the tumor, the carcinogenesis process is all about that there was actually a good intention for the stem cells to help. But some of the stem cells, hematopoietic stem cells, maybe mesenchymal stem cells, maybe oral stem cells, so we have oral mucosa stem cells. They might do the same thing we call stem cell niche defense mechanism. They employ this altruistic stem niche defense mechanism. That means temporarily suppressing p53 to gain some fitness. Some of the stem cells, if they do not delete themselves, if their oscillation does not come back to return to normal level, Then that became a foundation for carcinogenesis. Because having this low p53 as stem cells can immediately become a cancer cell. So that's kind of a things. Now, back to this carbohydrate metabolism now. So how, think about this, that if this is the case that as stem cells, the embryonic stem cells, or even we now found mesenchymal stem cell, has survived somehow, reduced p53, okay, I get it. I buy this, okay, if we finding p53 is deficient, but how do you explain that these cells are going to secrete glutathione? What is the metabolism? I mean, the cells just need to survive somehow. There is hypoxia, there is no nutrient there as well. So is there any mechanism they're doing metabolically? Is there a metabolic level? This is we call Warburg effect or aerobic glycolysis. But sometimes cells, like stem cells, even in the presence of oxygen, they actually do not use oxygen. They use lactic acid mechanisms. Here I will show you. I think you can find it here that from the pyruvate we can become lactate, so they do not go to the mitochondria. There's always a question in the literature for last 150 years. Warburg actually proposed this idea about 120 years ago. So why a cell decide to have aerobic glycolysis? For what? One of the examples could be that because it needs kind of to sustain in low oxygen like stem cells. Okay, one reason. The other reason could be that it needs to really synthesize many other metabolism from pyruvate lactic acid. I will explain why. Many of the hexose monophosphates shunt as part away all the nutrients for the nucleic acid synthesis. That's why. The other reason could also be because this is a defense mechanism. That when they are in an altruistic phenotype, they need to really only use this aerobic or anaerobic glycolysis now, because no oxygen, to really synthesize their machinery to shift from glucose synthesis, other carbohydrate metabolism, to either the glutathione synthesis or even isoprenoid synthesis, acetyl coenzyme A. You guys know that, those are students, that there is a relationship between all the metabolism. Glucose can become fat, amino acid, through different way. For example, acetyl coenzyme A produced from glucose metabolism, eventually and fat metabolism become diverted to produce isoprenoids through the mevalonate pathway. And I wrote about that in my book about squalene. So this is our question. And we have been studying about this, about the glucose metabolism, about lactate for last about, I think, nearly 7 years. And the data I cannot show you because, it's confidential. I am only showing those data we have published. But we're coming to a deeper mystery while looking for it. We are finding a combination between this isoprenoid metabolism and glutathione metabolism. So this is one of these things. I will talk about that, then I will go through rapidly so that our turn down, you can talk a little bit more. What's the time right now? We have 5 minutes, okay, yeah. So here, this is what we're doing, that from this pyruvate, there is the TCA cycle, there's a lot of regulation. PDK, we have snail, Heve, we have EMT, all involving in the tiny altruistic stem cells. And we're finding those mechanisms being hijacked by our cancer stem cells. So those patients who are dying, they are also altering the metabolism. And with this, I think I want to conclude that carbohydrate metabolism seems a key player in this altruistic defense mechanism, not only in embryonic stem cells, mesenchymal stem cells, also cancer stem cells. And if we can find these pathway inhibitors, because metabolism is a very important target for cancer with immunotherapy. So there could be a new discovery that is still working on. For those who are interested to visit our lab, we're welcome to see. We have a training program. Our Lekika partners, Lekika has been directing the training program for the last about 5 years. We have trained how many? 50? Yes. And hopefully also Sonja would be communicating between you and us, that we have a mouse model. With that, I finished my talk, and hopefully now we'll have time for Sonja and Lekika to briefly talk about our work. Thank you. Thank you.
Once it releases the glucose, does it sacrifice itself?
Oh yeah, those stem cells, they are going to die because after 2 weeks, the P50 is going to come back.
Because sir, in altruism behavior, one has to sacrifice, I think.
Yes.
Yeah, so that's why I was asking. Is that
Yeah. Okay, so what happened, as I said, the P53 MDM oscillation, the stem cells suppress, they start in a low P53 status state only for 2 weeks. During this time, they can secrete glutathione. After 2 weeks, this cycle come back, oscillation. Then this stem cell will undergo apoptosis. That's how they sacrifice their fitness. They did not persist, they did not continue to live. If they live, they become cancer cells. But that's why it's a sacrifice. Did I explain that?
Yes, sir.
Okay.
Not brief actually, because I'm from chemistry background, so whatever you have spoken to, it was really nice.
Yes, you are fair. Yeah.
So, like, yeah, I agree with you. Like government, I don't know who the policy makers, why they are doing it, because nowadays funds are getting reduced.
Yeah.
India is claiming that the economy is going up, like in next 10 years, they will be like second largest economy, or something like that they claim. But that's, what is the reason, or what do you think? That because after a few years, 5-10 years, I think state universities like us, it will be like just a teaching university. Because nowadays number of projects faculty members are getting is drastically reduced.
Yeah.
So in such a case, like whenever some like innovative ideas come to mind, we cannot execute. There's no way possible.
Yes.
So like, do you have any like what would be the?
Well, we have, I can say from my experience of a case study, I would say that like in 2019, we made an ethics committee for stem cell ethical research. We spent a lot of money. We invited specialists. We formed our committee, sent to ICMR. They're still considering. So they have developed a huge stem cell policy. The document is 200 pages probably. They're licensed rights. Before the idea comes, they come with their own policy. The bureaucrats. Guwahati Biotech Park, Sundanos, I can tell openly because I am not getting any government fund. So their CEO is an IS officer. It was started by our Manoniya Guha sir, what was his name? Ghosh sir. Anil Ghosh sir. He started, poor guy, he was a real scientist, right? He was kicked out by government also. The government assigned the control. How can an IS officer direct biotechnology? Fence, tell me. And then they have developed this BIRA. Get 50 lakhs. Tell me one thing came up from BIRA. Nothing. They gave us 2 grants for nanos. In the paper, they have given us grant. We did not get any grant. So this is a drama because these guys, they sit there, they go to international world or policy meeting. I do not know what they do. But when you talk to them, they're like our village small-scale politician with big egos. They do not know where the world is going. That's how they missed this whole biotechnology venture. They gave to China on a golden plate in 2001 too. They're doing the same thing right now because now Vietnam is going way up. Indonesia is going up, Malaysia is up. Singapore has actually more better technology. They have more powerful economy than whole India. Why? Singapore invested in stem cell research huge. It was not only about stem cell research, about understanding the molecular biology of stem cells, right? But Indian, stem cell means therapy. So they did not understand. And they did not read the grant. We submitted many cancer grants, never been selected. What they say, you grant something as which sentence they write. Not eligible, not something. Standard. There's no feedback. There's no feedback. And then there's a whole political lobby. They always get the grant. Right? So we thought that Modi government came to destabilize this lobby. We thought initially good, but they did not even destabilize, did nothing. So I do not know yet why. And I'm saying it, although I'm a part of Science Salon, I'm from Songha. Yes?
Okay, thank you, sir. So he's our international scholar, he's pursuing his PhD.
Oh, please.
Okay, thank you for your good presentation, it's very informative.
Thank you.
I would like to ask some question. Here, altruism, which is found in this human embryonic cell.
Yeah.
It's not known before this. Before this, your work, it is not that much known. This altruism is mainly common in bacteria. So now, when we relate this altruism to cancer,
Yeah.
as a result of developing this behavior of altruism, cells may mutate, leading to cancer. Now, is altruism important for cancer treatment or it leads to cancer? That's my question. This altruism, it leads to mutation leads to cancer. Now, how we can exploit this discovery of altruism in human cells for treatment of cancer? That's my question. The other is, we have talked about carbohydrate metabolism. What's the need of converting carbohydrate into glutathione, and what it is impact on these cells, stem cells, which can lead to mutations? Or is it enhancing this process, or is it reversing this process? What is the impact of this glutathione on this role of converting these stem cells into altruism type of cells? That means cells that can produce different molecules that can impact the environment. That's my
Thank you.
question.
Good question. Where are you from? Which country are you from?
Ethiopia.
Oh, nice country, Ethiopia. Yep. Nice to see Ethiopian scholar here. The first question is, can you take advantage of this altruism that is probably that cancer is hijacking, right? So yes, we have a manuscript we published. Our other PhD scholars, Sima Boya. You can actually Google BCG vaccine that can induce altruistic cell-led to cancer. What we did, certain microbes can go to the cancer stem cell, exploit this altruism. So fundamentally what I would say is that since altruistic stem cells are deficient in p53, and cancer also has p53 deficient, if you can return the oscillation, p53 and GM to oscillation in cancer cells, can we induce an altruistic cell death? We have defined that altruistic cell death. It's a whole manuscript. And once we end this meeting, I'm going to ask Lakhika to or Sandhya to send a manuscript. We'll read that, you'll find the answer right away. Okay? So, next question is about glutathione metabolism. That if the glutathione metabolism is now contributing to the altruism, so how is it linked to the carbohydrate metabolism? That's the question, right? Yes, this is where what you're finding a link between glutathione and isoprene metabolism or squalene metabolism. I will ask you to read one of our first paper on this, published in 2003 in European Journal of Cancer. So 2003 we showed a link between glutathione metabolism and squalene. I'm going to also email this paper to you. Suffice to say, because we cannot disclose many things here, that there are very atavistic metabolism that is inherent in bacteria. Like glutathione or isoprene metabolism also come bacteria. And this is where also we're studying this Mycobacterium tuberculosis. I didn't talk about that because we published a paper in Science Translational Medicine showing bacterial stem cell crosstalk. We're the first lab to show in the world that bacteria can live inside stem cells. So we're finding this metabolism is pathogen-host interaction. It's also important. In fact, recently, we have a manuscript would be submitted, Bharat is working on, we're showing how bacteria and cancer stem cell crosstalk. But because of funding issue, our lab got delayed. So even though we were the pioneer in the field, because we came to India, we could not position ourselves. So now this microbe cancer field has moved out big. And Hanahan, he has now included microbiome as another hallmark of cancer. So we're showing that, even though we missed the boat, but we're the first ones, we can come up. But my point is this, you have to ask a big question on that. Metabolism in cancer, we're finding glutathione metabolism, carbohydrate metabolism, isoprene metabolism. If you look at a textbook, you see no relation, nothing, the distinct metabolism. But we are finding the link, because at the end, all the metabolism come from a core metabolism in a primitive unicellular life. And we are finding this atavism. Atavism means reappearance or evolution of certain deep historical evolutionary mechanism. This is what we're finding in altruistic cancer stem cell, a lot of novel metabolic patterns. One of them is glutathione and isoprene metabolism. Thank you.